Hello, I'm Jeff Meggitt. This is the first of two short videos that act as an introduction to my book Genes, Flies, Bombs and a Better Life. In it, I tried to weave together the history of genetics and the life of one of its key figures, Hermann Muller. The first video is a much compressed and so partial and incomplete summary of developments in genetics to about 1900. The second is much more about Muller's contribution and his extraordinary life. You can skip the first one if you know something of the history. You can skip both and just read the book. It's available on Amazon, and there are more details on our website, pitchpole.co.uk. People must have noticed family likenesses and traits long ago, but they were generally mysterious things appearing and disappearing with the generations, touching some offspring, skipping others. Inheritance was inextricably linked with reproduction, and some understanding of how it worked had to wait for the Greeks, and Aristotle in particular. He studied the development of chicken eggs and concluded that the embryo and its organs developed from the tissue in the egg, a process called epigenesis. In higher animals, the mother and father somehow both made contributions. In mammals, the menstrual blood and semen each, somehow, drawing characteristics from the whole bodies of the parents. They combined. It was an idea that was to be revived in the Renaissance and persist until the 19th century as pangenesis. Charles Darwin favoured it. Little real progress beyond an appreciation of the Greek achievements was made until the changing attitudes of the scientific revolution and the invention of the microscope just before 1600. In an age of enthusiastic dissection, this revealed not just fascinating external details, but also complex internal structures and the development of insects through the larval, pupal and adult stages. It showed the sperm wriggling through the seminal fluid. Some thought them parasites, but they were soon seen as the agents of reproduction. Spallanzani's beguiling experiments in which he fitted male frogs with well-tailored gingham trousers and found that they prevented fertilisation is often quoted, perhaps misleadingly. An alternative explanation of inheritance, pre-formation, held sway for a long time. This claimed that each offspring for all future generations existed right from Adam and Eve, perfectly formed but absolutely minute in either the egg or the sperm. It may be difficult now to see how this could be credible, but in various forms it was accepted. But then the creationist idea of the chain of being was widely accepted too, certainly up to the middle of the 18th century. This organised creation into a series of layers, with God at the top, then angels, then humans, then apes, and then layers of other animals. Plants were somewhere below that, and then there were rocks and other inanimate things at the bottom. The chain had plenitude and continuity. Plenitude because the Creator, being perfect, had already created everything that should be. And continuity because everything had to be neatly connected. So species were fixed in this scheme, and hybrids, and by the end of the 18th century many were known, were seen as a real challenge. The discovery of the sexuality of plants in the 18th century was a great shock to believers and provoked outrage. Plants, with their multiple sex organs, seemed to be having too good a time. Science nibbled away at the chain. Fossils were a particular problem. But the creationist interpretation was hardly challenged publicly until the 1840s. Charles Darwin, at the middle of the century, confronted it in his Origin of Species. The careful work of a Silesian monk, Gregor Mendel, in the 1850s and 60s, probably directed at resolving questions over the fixity of species, marked the beginning of the science of genetics. Mendel studied the inheritance of several well-defined traits in peas and found that the pattern of this could be explained if there were two factors determining the trait, one inherited from the mother and one from the father. One of Mendel's traits was pea colour, where it seemed that these two factors could come in two forms. Let's call them green and yellow. If a plant inherited green factors from both its parents, then all the peas would be green. If both were yellow, its peas would be yellow. The peas of a plant having one green factor and one yellow one might have been thought to be greenish yellow, but in fact they were always yellow. Yellow was the dominant factor and green the recessive. The other striking result was that inheritance of the factors was a random process. A parent with a green and a yellow factor would pass on one or the other to its offspring with equal probabilities. Mendel's results could have been an important insight in the search for the physical basis of inheritance, but, alas, although published in 1865, it was hardly known until rediscovered at the turn of the century. The study of the cell became more fruitful from the 1830s as microscopes gave better images and preparation techniques improved. It was recognised that all plants were composed of cells, and the discovery of mitosis followed soon after. The internals of the cell outside the nucleus were being studied by the 1870s. There was good reason to think by the 1880s that the key to inheritance lay in the nucleus, and by about 1900 there was strong evidence from the work of Sutton-Boveri that the newly discovered chromosomes were the carriers. Mendel's work strongly supported this when rediscovered. The behaviour of the chromosomes was consistent with that of his factors. Important in the 1880s was August Weismann's promotion of the germplasm theory. This had it that heredity worked solely through the germ cells and that these were unaffected by the other cells of the body. There was what was called a Weismann barrier. It was Walter Sutton, working at Columbia University in 1902, who produced clinching evidence from his work with grasshoppers, clinching for many anyway, and a truly coherent theory of the role of chromosomes in heredity. It's become known as the Sutton-Boveri theory. While this provided a theory of Mendelian inheritance, there were several issues that it did not address. For example, it did not explain quite how non-discrete traits such as height were inherited, although it was soon to be adapted to do so. It did not explain why some factors were dominant and others recessive. And so we arrive at the new century, and the years around this mark three key events: the emergence of the USA as the real driving force of much of genetics research, the start of the career of a tiny fruit fly, Drosophila melanogaster, the black-bellied dewlover, as a superb experimental subject, and just a little earlier, the birth of our human subject, Hermann Joseph Muller, in New York in 1890.